Hi, we are Group 2, and we are discussing Avery's experiment. So, before we get into Avery's experiment, we have to look at the experiments before. So, we'll look at the Griffith experiment. Griffith performed this experiment in 1928, and it was an experiment that proved transformation existed. Griffith found that when you add heat-killed virulent smooth cells with living non-virulent R cells, there's a genetic transfer between the two. The R cells grow capsules and become S cells, and when injected into mice, the mouse contracts pneumonia and it dies. So, years later, Avery conducted similar experiments. So in 1944, Avery wanted to find out what is the genetic material that is passed between heat-killed S cells and living R cells. In order to do this, he performed 3 experiments. One added DNase, one added RNase, and one added proteases. DNase, when added to the mixture, breaks down DNA. When RNase is added, it breaks down RNA. And when proteases are added, they all break down different forms of proteins. What Avery found when performing a similar experiment with Griffith, when he added either RNase and the times that he added proteases, the R cells were still able to get genetic material from the heat-killed S cells, were able to form capsules. The mice that they were injected to contracted pneumonia, and the mice died. But he found when he added DNase, the R cells were not able to get genetic material from the S cells. Therefore, the mouse did not contract pneumonia, and the mouse lived. It's ugly. So therefore, Avery found that he was one of the first to prove that DNA is the genetic material that is transferred between heat-killed S cells and R cells.